Students, I'm Udwa Allen, your facilitator for Bio 216. In this video, we'll learn about nucleosides. Listen and take notes. Thank you. This is Bio 216, Chemistry of Carbohydrates, Lipids, and Nucleic Acids. We have so far dealt with some aspects of the carbohydrate lipids. Now we're looking at the nucleosides. And to start the nucleosides, the nucleic acid, we have to consider some of the parts of the nucleic acids. So today we'll look at the nucleosides. At the end of this lesson, you should be able to explain what nucleosides are, list the different components present in nucleosides, please, and you should be able to identify the structure of different types of nucleosides. What are nucleosides? If you look at nucleic acids we have the DNA and the RNAs. A monomer of each nucleic acid is composed of three different things: nitrogenous bases, pentose sugars, and then phosphate group. So when you have the association of the nitrogenous bases and the pentose sugars alone, they are called nucleoside. That's the association of the nitrogenous bases and the pentose sugars are called nucleosides. And for this nitrogenous bases and the pentose sugars to be associated or linked, it is made possible by a type of bond known as N-beta glycosidic bond. I hope we're not going too fast. I'm sure you'll be wondering, what are nitrogenous bases? Because I believe by now you already know what pentose sugars are. So let's start from somewhere. Of this purine, you can see, I want you to note the way these things are numbered. You can see where number 1 starts. Each of the atoms of this structure has its own number. You don't just number them anyhow. And you note, see where we have N represents nitrogen. Then when we don't have anything, like number 2 atom, it's a carbon. It's carbon there. Number 4 atom is carbon, 5 carbon, 6 carbon. Whereas 7 and 9 are nitrogen atoms, then 8 carbon. Same goes to this pyrimidine. Number 1 is nitrogen, number 3 nitrogen, then 2, 4, 5, 6 are carbon atoms. So if you see now, this, like I said, is the backbone. So what we adenine is formed from the backbone, the backbone of the purine. You can say they have the same structure, but they now have additional atoms that differentiate them from one another. For adenine, there is an amino group attached to one of the carbons, one of the atoms, which is what? Which atom is this? Yeah, now check and tell me which atom it is. Good. That is carbon 6. Atom 6 has a nitrogen, an amino group attached to it. And then for the guanine, you can see that the atom 2, which is also a carbon, has an amino group attached to it, in addition to atom 6 having an oxygen attached to it. And you can see, because of this attachment of oxygen, a double bond that was present here has gone to form this group. So you see, these are the differences between adenine and guanine, and it's good you note. So when you see a compound formed from adenine, you will know, and that formed from guanine, you will also know. So let's look at the pyrimidines. Which of these will give us the cytidine? Good. Cytosine. Good. So we'll take this cytosine and add it to, link it to what? What are we going to link it to? Is it this or this? Good. Ribosine. Link it to a ribosine, is cytidine we're drawing. So what do we have? Try it out. Score yourself. So at the end of this lesson, you have been able to note that, note the following. Good. So thank you.